Hello students, welcome to Dr. Deepa's NEET Biology, and in today's session we go through important tables of class 12th entire syllabus, and these tables are most to be revised for final revision by NEET aspirants. So we begins with chapter 1, and one by one we come towards each and every chapter that what are the important about these table. Now here you appear the table 1.1 from the chapter 1. In this table, mainly the informations are provided regarding chromosome number in meiocyte. All you are now knowing what is meiocyte, and meiocytes are the mother cells, diploid cells. So you clearly appear the column. Fill in the blanks are provided here. This is actually work for students. So now we are not going towards other formal part because this is the only revision, super fast revision here. For final revision, one time pay attention on the chromosome in housefly, chromosomes of Ophioglossom in fern, as well as the chromosome number in fruit fly, because these three examples are repeatedly asked in previous exams. So this table, one time you go through this table and very well prepare this table for your final revision of NEET exam. Now we come towards the table 5.1. This table is of from chapter 5th and page number 71. In this table, that you all the students very well I think prepared this table, but one thing you miss that you prepared this table in terms of the shape and in terms of the color. So one time you go through the table again and appear the table that how many characters are used for the shape. So if you appear the characters for shape, so mainly two characters we studied regarding the shape, that is the pod shape and seed shape. Second question generally asks that how many characters contrasting trait of a Mendel's are related with color. So with color you remember three traits are there: flower color, pod color and seed color. And in this way you remember one another question is asked already, that how many traits are used by Mendel which were related with flower. So with flower you remember only two traits are there. So instead of preparing, you already prepared, I know that the contrasting trait you prepared, but one time you revise this table in this way also. Now we come towards the table, next table. That table is also of the same chapter. The table number 5.2, page number is 77. This table is showing the genotypic basis or genetic basis of blood group in human population. And here you mainly pay attention on the blood group AB, that blood group AB is example for codominance, and for this table you very well prepared that there are 4 phenotypes of blood in human, and for 4 phenotype 6 genotypes are available. If you go through the table, so you clearly appear 7 columns, but these 7 phenotypes are not there because for blood group AB the common phenotypes are present, that is IA and IB, and that was the one common genotype. So this part you remember. For AB, though you appear AB in this chart for 2 times, but if you come towards the genotype, so IA and IB is the genotype for blood group AB. So this part you remember very well, and again for objective purpose you remember for 4 phenotype of bloods only 6 genotypes are responsible. Now we come towards our next table, that is again one of the most important table for the statement based questions. Table 5.3. This table is also from chapter number 5, and you appear this table on page number 82. When you appear this table or you go through table, so the table is mainly explaining comparison between chromosomes and the behavior of genes. And when you finally revise, so you very well revise the third point. You pay attention on the third point of the comparative table, because this is the point which make a difference between the chromosome and behavior of the genes. Now we come towards the table 9.1, that is table for chapter 9, page number 174, and this is one of the most important table. And in this table you mainly appear number of crop plants and their varieties are developed. The varieties are developed mainly किस लिए डेवलप हुए हैं ये varieties देखिए. The varieties are mainly developed for the resistant to disease. So जब आप ये फाइनल रिवीजन कर रहे हैं, सो ड्यूरिंग फाइनल रिवीजन you go through this table, and mainly जो cauliflower, you prepared the variety of cauliflower very well, that is Pusa Shubhra and Pusa Snowball K1, that you remember very well. And second most important is Pusa Sadabahar. So for what kind of diseases these varieties are resistant, that you prepared from this chart. Entire chart is important, but Pusa Snowball K1 and Pusa Sadabahar is again one of the most important. Now the next table हम discuss कर रहे हैं, that table is 9.2. सेम चैप्टर से आपका टेबल चैप्टर 9, एंड टेबल इज प्रोवाइडेड फॉर प्लांट ब्रीडिंग यूज फॉर डेवलपिंग रेजिस्टेंस टू इंसेक्ट पेस्ट. And when you go through the table, so number of varieties and number of three number of crops and their different varieties you appear. The most important is Pusa A4. So while final revision, you one time revise properly Pusa A4 for the what kind of insect pest this variety is resistant, and as well as in addition to that you remember Pusa Gaurav. Pusa Gaurav, you remember it is resistant for the aphids, and it is a variety of the crop mustard or brassica, you remember. Now we come towards the next table. That next table is actually no number is provided after chapter 9. In 10, 11 and 12 no table is there, but in those chapters a large number of diagrams are provided, so that we will discuss separately. And here we come towards one of, you can say, table. I use that diagram as a table because this is one of the most important. This diagram you appear on page number 229. Most important diagram है, repeatedly asked this, repeatedly पूछा जा रहा है. Secondly, one equation also provided in NCERT just below that diagram. वो इक्वेशन भी आप प्रॉपरली प्रिपेयर करिएगा. पेज नंबर 229 पर है आपका ये. एंड दिस टेबल इंडिकेटिंग द इफेक्ट ऑफ 4 फैक्टर्स ऑन द पॉपुलेशन डेंसिटी. किस तरह से पॉपुलेशन डेंसिटी आपकी इंक्रीज होगी और डिक्रीज होगी. प्लस एंड माइनस साइन के लिए भी आप याद रखिए, क्वेश्चंस पूछे गए हैं. So plus sign, you remember, indicate the increase in population density. Minus sign show the decrease in population density. Diagram based question के लिए one of the most important aspect है. So you also prepared final revision के लिए आप ये diagram भी बहुत अच्छी तरह से prepared कर लीजिए. So these are the some tables of the class 12th, and then from ecology section we come towards the next table. In ecology, you predominantly appear the 2 tables in pollution control. So in pollution control, one table you appear, that table is 13.1, and that table you remember is mainly for the population interaction. This is also one of the most important table. In the secondly, plus minus, what does it stand? नेम ऑफ द इंटरेक्शंस भी पूछे जा रहे हैं। तो ऑलरेडी अब आप लोग सब कुछ सॉल्व कर चुके हैं, घबराने की ज़रूरत नहीं है, खुद के ऊपर फेथ रखिए, पेपर सभी स्टूडेंट्स का बहुत अच्छा जाएगा। बट सम ऑफ द स्टूडेंट रिपीटेडली पूछ रहे थे कि व्हाट आर द टेबल्स आर इम्पॉर्टेंट, कौन से डायग्राम्स में फाइनल रिविजन के लिए, लास्ट रीडिंग के लिए इम्पॉर्टेंट है। सो दैट्स वाई आई प्रिपेयर्ड such videos and provided to you। तो यहाँ पे 13.1 टेबल आप बहुत प्रॉपरली प्रिपेयर करिए, एंड आफ्टर दिस 13.1 टेबल one another table is provided। that table you appear on the screen, that is the table for chapter number 16, and now we come towards the table 16.1। 16.1 टेबल जैसा आप अपियर कर रहे हैं, इट इज दिस टेबल प्रोवाइड किया गया है आपको मेनली फॉर मास इमिशन स्टैंडर्ड्स इन इंडिया। नॉर्म्स जो है वो इंडिया में भारत स्टेज 4 है। सो भारत स्टेज 4 और यूरो नॉर्म्स 4 हम कह रहे हैं, उसके लिए 4 व्हीलर्स, 3 व्हीलर्स एंड 2 व्हीलर्स के लिए। सो यू ऑल्सो गो थ्रू दिस table for the chapter number 16, and this much tables are very important। so one time you go through all the table, hardly 15 or 20 minutes are required, not too much time, because already सब कुछ आप लोगों का prepared है। finally किन points पे important and stress provide करना है वो आपको यहाँ पे बस थोड़ा इंडिकेट किया है। तो टेबल को वन टाइम आप उस पॉइंट ऑफ व्यू में रिवाइज कर लीजिए, एंड अप टू नेक्स्ट सेशन स्टे मोटिवेटेड, स्टे ब्लेस्ड, एंड स्टे कनेक्ट विद द डॉक्टर दीपा डाके's नीट बायोलॉजी।